So my presentation, I seem to have lost, just a second, we lost the. Okay. So I'm going to talk about skin sample genetic preservation and cloning for conservation. So I'm going to introduce Gemini Genetics and our partner Biogen Pets and Equine to you, and then outline skin sample preservation, the applications and benefits of working with skin, equine cloning, cloning for conservation, and then our latest area of work, which is for the UK National Livestock Biobank. So Gemini Genetics is by heart a skin sample genetic preservation company. We're preserving DNA via skin samples for future use. The main outlet of that being Gemini is a company, it's a business, so our main commercial outlet of that is for cloning. We work across horses, but also cats and dogs. Dogs actually being our most common sample to be submitted to us for commercial cloning. Our founding, though, is from Rare Breeds. So I used to work at Stallion AI Services. I still do jobs on the side for them. But I used to be a lab technician down with Pam in the Stallion lab, started in 2017, processing the semen. And Tullis, very much his work involves supporting Rare Breeds, as you learned all today. He went to a conference in 2018 and came back with the idea of, can we use skin for anything? Because we're working to bank semen down from these animals. And that's brilliant, but that's not preserving the mares. And somebody has already said today about the issues that we have in we can't actually preserve mare eggs. Their membrane is too hard for the chiroprotectant to go in to protect them during freezing. They've also got quite high fluid content. So they burst, they rupture when we freeze them. So how can we preserve our mares? And Tullis came home with this idea from the Livestock Conservancy presentation about whether we can use skin, and really said, can you do something with this? And that's really where Gemini Genetics came from the want to preserve mare lines. And from there, we've branched out to other domestic animals too. But very much the founding of us is in rare breed preservation and wanting to preserve down mares. So as a business, we're partnered with Biogen Pets and Equine. They are a US-based company. They're based in Texas in the US. And they are a fantastic team of people that also have a number of arms to their business. So yes, they are commercially, in order to be a business, they are a cloning company, but they also do a lot of work for conservation, and they're actually working predominantly with US-based zoos and the USDA, so a bit like the DEFRA equivalent in the US, to do biobanking for zoo species conservation in addition to their commercial cloning. So we visited them in January to see their facilities. They had an update to the facilities, so we went over to view that. And another part of their company is actually for ICSI, intracytoplasmic sperm injection, injecting sperm into egg. Because you'll see the way that cloning is done is not too dissimilar from ICSI, and I've got some videos to show you on that later. So the first stage, myself and Amelia were showing you during the practical sessions, the genetic preservation. So the first stage is we receive skin samples from animals. And our first step is to preserve the DNA within that skin sample. And it's very comparable to semen freezing there, where we're wanting to essentially put the DNA to sleep so that it's not going to degrade and we can wake it back up in the future. So it's a combination of using the correct freezing media, the correct extender, and also the correct freezing curve. So we don't just take these samples and put them straight in the freezer. They go through different increments of temperature to enable them to freeze successfully. So that's stage one. You can take the skin sample, you can have it banked, and it can be sat there the same as frozen semen to be used in the future for whatever purpose required. And in terms of the samples that we need, if it's from a live equine, we request 4 skin biopsies, 6 millimetres. The diagram, they're not that big, it's just we need to be able to show you on a picture. It's only 6 millimetres, it's done under standing sedation and local anaesthetic. And for a post-mortem equine, we asked for those neck punches plus a small section of skin from the ear, which we were showing you earlier. And one of the key benefits of using skin samples is that we have up to 5 days to receive the sample after it's been taken or after the animal's passing. So that's a very good, more leverage on time versus testes, which we've learned today. We need to get them to us as soon as possible. We work with skin because it contains fibroblast cells, and those are the cell type that we are culturing and ultimately going to use for conservation of these breeds. And in terms of biopsy punches, it is just the skin we're interested in. So we don't want a deep punch going into muscle and fat tissue. We don't use that material. We're just using skin. And I think Amelia highlighted we have to have the samples sent to us chilled, not frozen. So if you stick a skin sample straight in the freezer, the rungs of the DNA helix, they're going to be damaged by that. And so samples we kept chilled and sent within 5 days. And we do have a number of videos on YouTube that offer guidance on sample packaging. It's also in your booklets as well. So the benefits of using skin samples and of preserving skin is firstly, it contains the whole genetic profile of the animal. So sperm and egg just contain 50%. The skin sample contains the whole genetic profile. And that whole genetic profile may be key to the future. It may hold genetics that we need to capture in the future, and it may be key to that animal's certain resilience or trait characteristics that we're looking to recapture. Another key aspect, which somebody asked me, I think, during the practicals, was that it enables us to preserve across all genders. So we can, because we're working with skin, we're not reliant on semen being present. So we can collect from geldings, we can collect from mares, we can collect from those that are not sexually mature or those that have gone past optimum reproductive quality. The other aspect of skin samples is that it's an infinite resource, and that's a really important one. So when we've got a bank of semen, if you collect 100 doses of semen from your stallion, each time you use that semen, it's going to go to 99, 98, 97, carry on, as you inseminate mares. The skin samples, though, we spoke during the practicals that we culture the DNA. And during that culture, we actually multiply up the genetic resource. So we start, for example, with 1 vial of preserved skin, and we culture it, and we generate 4 vials of cells. So we've gone up by 4 just in that one generation. And then each of those 4 vials of cells can in turn be multiplied up. So it provides a more infinite resource for use for conservation in the future. And also has the possibility of being easier to adapt to future requirements. So if we are looking, rightly or wrongly, into an area where we might need to look at genetic modification of animals, whether that be to provide resilience to diseases we can't control by other means, whatever requirements that is, the skin samples might be easier to reprogram for those requirements in the future. There is also less variability in the viability of skin versus gametes. So if you've got a really well-taken skin sample, it arrives within 5 days, it's been kept chilled, we should have a 96% chance of being able to create and preserve living cells from that skin sample. Relative ease of sample taking versus using sending stallion to us or semen collection, and you have got that 5-day window for shipping. So in terms of how application number 1, cloning, is done, sorry, it's jumping itself. The process works by we take a donor egg cell of the same species that you're wanting to clone, and we remove the nucleus from that donor egg cell. So then it becomes a hollow egg. We then take a skin sample from the animal that we wish to clone, and we culture the DNA in the laboratory. We then insert one of the skin cells that we've cultured in the laboratory into the hollow donor egg cell, apply an electrical impulse to that, which mimics what happens during fertilization, and that egg is then allowed to mature for 7 to 10 days in the laboratory, making sure it's going through the correct embryonic stages before being transferred to a foster mum for the pregnancy. So these are some videos here. So this is the first stage of the donor egg cell having the DNA removed, so that it becomes the hollow egg ready to input the DNA of the donor. And step 2 here these are the skin cells that are being collected up. They're circular in appearance, which is different to what you saw during the practicals. This is because these are not attached to the bottom of the flask. So when we detach them, they curl up a bit like when you catch a spider, it goes into a ball. The previous images you saw was when they were in cultures, they were attached to the bottom of the flask. So they've gathered them up there into the needle, and they're now fixing the egg in position, and they're going to implant the genetic material in. And you can see that's very similar to the video that Petra showed earlier in terms of ICSI, so injection of the sperm into the egg. So for horses, cloning actually isn't too dissimilar from what we're already doing quite extensively now in terms of ICSI, and they do a lot of this ICSI at Biogen as well. It's just instead of injecting sperm, we are injecting a skin cell. And we have 2 cloned animals here. So I don't know if you've met Jem. Is Jem here today? Jem Dog?
I don't think so.
Okay, maybe need to request Jem Dog to come over here. So we have our own cloned dog called Jem, Cocker Spaniel, obviously the one nearest me. So she's a lovely little girl who started her life in our laboratory as a section of ear from the original when it passed away. So that's Jem, and we'll actually make sure she can come over here today to meet you all. And then we have Horse Jem. They were named separately, but it just so happens they ended up with both the same name and similar to the company. So he's Mercus Jem, and hopefully you saw him today. Yeah, when you did your garage tour. Yeah. So Mercus Jem is a clone of a gelding. So Jem Twist, number 1, is the original. He was a US-based show jump back from the 1980s region. And Mercus Jem, our stallion, is one of 2. There is another clone called Gemini that stands in France. And Mercus Gems at a really exciting point in his career now, where we've got the offspring on the ground, they're out in the competition circuit. So you're seeing the carry on through of genetics. In the US, cloning is just very much considered a standard thing. There's loads of examples of cloned horses in the US. These are just a few from the UK and Europe. So there's Cruising, Irish-based show jumper. His clones being Cruising Encore and Cruising Irish. Chilly Morning, if you're eventing, you may be aware of, they've got 3 clones from them. One with Gemma Stevens, one with Alex Hugh-Tan, and one being kept for breeding. There's also Tamarillo, so we've got the original and the clone there. And a relatively recently announced one is Pacino, another Irish stallion. So an intermediate stage, once we've preserved the DNA, is to culture it. So again, we showed this during the practical. And thank you to Yvonne for letting us use the photos from Robin's cell culture. So this is where we take the section of chunk of skin that you saw in the Petri dish. We add an enzyme to it to digest it, so that it releases the individual cells that make up the skin. And we then aim to culture them, so increase them in number. So these are the fibroblast cells that you saw during the practical. These all have the nucleus containing the whole genetic profile of the animal, and it's one of these cells that are injected into the donor egg to become the clone. When samples are submitted, we also work with animal genetics if the owner is happy to do so and submits a sample. Most commonly with hair being easiest for horses, we send them off to animal genetics for an independent DNA profile, that being used obviously for verification in the future of those samples, whether that be matching up to the clone or other technologies. Why do people clone their animals? So we do, as I say, dogs are our most common client base, but we do work with a number of rare breed societies across equine and canine as well. So we had the equivalent of this day, but for dogs last year, and that was a big success in highlighting the plight of a lot of rare breed dogs and also the use of this technology. As a mainstay though, there's quite an emotional perspective behind it, to maintain a bond with a much loved animal, carry on a legacy and a connection to an animal. People's cats and dogs tend to be neutered, so it's a bit different to our horses where we can, we're maybe breeding off a mare that we really love and that's going to be our carry-on. Commercial cloning of an elite animal, and then obviously it's conservation that we're talking about today. 1 point on our record keeping and database management. So actually the management of the database is as important as the samples that you're processing. So you want to be managing a database that ideally in the future other people are going to be able to look at that to know what's banked, see the gaps, see the resources available. We work with a human IVF system for this. It's called Matcher, IMT Matcher. It's very much future-proofing system because it allows to be interconnected to other databases. All the samples are barcoded as well, which will allow for independent witnessing. So they've got these mini robots that can independently witness that I've brought the right horse out of the tank to do the culture. So it's very much future-proofed and set up for future requirements. Storage, obviously, also important. So we're aiming to store the DNA across multiple tanks so that all your eggs are not in one basket. All of our tanks are alarmed. You would have seen that today. Fireproofs, rooms, CCTV, etc. And one end goal would be to have them in multiple locations. So we do, at present, upon owner's request, we can split the samples between Gemini and Biogen. So as well as cloning, another application of which we're working towards is being able, a future where we might be able to generate sperm and egg cells from the preserved skin. So we're working with a lab in Switzerland at the moment for this. So this has been achieved in lab-based mice, but they're often species where we can sometimes achieve things that we can't then achieve in other species. So we're working with a group in Switzerland that are slowly moving away from that set of animals onto more potentially challenging types and seeing if they can carry through the technology. But the first step is to go from having a skin sample and converting it to an induced pluripotent stem cell, so a cell that has the potential to become other cell types. And from that stage, we then want to diverge them into egg or sperm. So this is something that's very much in progress, but it's another reason why we need to be banking down genetics for if this future technology becomes commercially available. In terms of cloning for conservation, we need to be preserving individuals that are representative of the breed or species, to protect the breed or species from challenges, and also to enable us, if we need to recall lost genetic diversity. Andy pointed out we're obviously breeding within closed populations, so it may be a very useful tool for if we're banking DNA now, that we may be going to look at using in 10, 20, 30 years' time to actually recall some lost genetic diversity. So an example of the use of cloning in conservation is from our partner Biogen, working with San Diego Zoo and another group called Revive and Restore, looking at the Przewalski's horse. So this is a horse that is extinct in the wild. The population today is captive bred. There's about 1,900 alive today, but they're all descended from 14 founders that were wild caught between 1910 and 1980. So they've got a narrow number of founders, and they have concerns about the level of inbreeding in their population and the risk of that to the population fitness that we've spoken about several times today, about the risks to reproductive health, increased risk of deleterious alleles coming through, etc. But thankfully, San Diego Zoo, which we also managed to visit in January, they had the foresight to begin banking samples as far back as 60 years ago, banking down skin samples from deceased individuals. And at that time, they didn't know what that was going to be used for, but they just banked them just in case they became useful in the future. And they apparently have over 400 samples in store for this species alone, and oldest sample from an individual born 68 years ago. So their use of this technology, they basically analysed the samples in storage and the current population, and they identified individuals that were least related to the current population and reintroduced them via genetic replication or cloning. So this is Kurt. Watch this video sometimes, please. Oh, there you go. So he's going to jump a bucket in a minute. There you go. So he's the world's first cloned Przewalski's horse. He is a genetic replica of a horse that existed over 40 years ago. So over that time, the population, the living population genetics have changed quite a bit. So Kurt is now, he brings back a lot of lost genetic diversity that they've had over the years to that population. He is now a 3 coming on 4-year-old, so he's about to go into the breeding herd. So we're very much about to just see the benefits that he brings. And they now, I haven't actually got a photo of him, but there is now a second copy of Kurt. He is called Ollie. He's much younger. He was only born last year, I believe. So he's got a little bit of time before he's out in the breeding herd. But this line was considered so important that they've made 2 stallions essentially to go out into the breeding herd. And they've now also started to work across other species. This is the black-footed ferret. She's produced from a skin sample that was preserved 30 years ago, and they now have an active program in place with the USDA with funding. They're taking skin samples actually from live wild caught animals of certain species to get them banked, get the cells cultured, and actively start utilising samples in this way. So when you wanted to bank samples, you wanted to capture animals that are essentially the pillars of the population. Who are those that are valuable, holding up the broad range of the population that exists? And one key thing about the the native breeds as well is that they are true to the environment that we've got here, and that's a really important characteristic that we need to capture. And it's actually something we need to consider when we're managing them. You know, we need to be still allowing them exposure and development within the climate, within the environment that they're designed to be. We don't want to start interfering with that too much, because then we'll lose their adaptation to the environment that they've grown up in, and that's the key aspect about them. So this is just a diagram to show, visualise, breeding again within a closed population. And if we're able to collect what's available to us today, and then that population goes through a narrowing in genetic diversity for whatever reason in the future, we can look back at something we banked 20, 30 years ago, look at genetically replicating them, and in doing so, bring back a lot of lost genetic diversity without the need for outbreeding or crossbreeding. In terms of how similar the clone and the original will be, they are genetic twins, so they have the building block to become the same as the original animal. We do need to remember the influence of the environment has the ability to change the characteristics of an animal. So they start off with the same building blocks, but you need the same input for optimum closeness to the original. But for conservation, we're mainly looking at focusing on the value of the genetic twin. And in terms of costs, there's a breakdown in costs in your information packs. We do offer the genetic preservation for rare breed horses at a reduced rate of £500 for that. That's different to commercial. And sort of guide price for cell cultures there, but we are flexible. Also, it depends on if breed societies want to work on banking a number of animals at one time versus one coming in individually now and then. The price could be different for that. So there is flexibility in the pricing available for conservation. Common question asked is, obviously the cloning at the minute takes place in the US, will it become available in the UK? Especially if some of these samples might be submitted with domestic use only, you know, if an animal's passed away, we've not had time to take blood samples. So there is, the EU is, cloning in the UK was banned under EU legislation. Now we've left the EU, there is potential for that technology to become available in the future. And we're already identified as a stakeholder for when they start to look at precision breeding in animals, which they've given us a rough date of 2026 by then for those discussions. I don't envisage that cloning is going to be something that comes through immediately, but I view it more as there's options for that now. And not having it here in the UK, there is a demand for it, and at the moment we're exporting it, and we're devoiding ourselves of opportunities to fully utilise this technology. And so it is an important one to discuss. I just wanted to finish now just on the last section of the latest venture that we're working on. So Tullis is going to speak later on in the day about Nature Safe. This is a wild and endangered biobank, and spoken today about how we can use skin sample preservation, semen, etc. to support domestic animals in terms of horses and cats and dogs if you're going down that route. But the missing piece in the puzzle is our livestock, the branch that we haven't covered. So we've recently launched, and it was announced on Countryfile a couple of weeks ago, the UK National Livestock Biobank. So this is a biobank that's very much based on the principles that we're trying to apply to rare breed horses, to wild and endangered animals, etc., but for UK food and breed security. So the reason for this is that our biobank, our livestock and food security are at risk. 75% of the world's food comes from just 12 plants and 5 animal species. So our food system is propped up on quite a narrow number of plants and animals. The FAO predicts that up to 30% of livestock breeds could be at risk of extinction, as many as 6 breeds lost each month. And part of the reason for this is that our world is changing. The environment that we're raising these animals in, with climate change, increased risk of disease, all these challenges place a lot of pressure on the food system. On top of this, our livestock are not sufficiently protected. There's not one single place we could go to if there was a large decimation of our livestock, through a disease outbreak, for example, to repopulate and recover them. So we could have irreversible loss of genetics at the herd and species level. And this is actually identified in the UN 2030 agenda in their assessment of goal number 2 to end hunger. They analysed local breeds and transboundary breeds, and there's only 4.31% of total local breeds and 8.83% of total transboundary breeds that are banked in a way that if there was a significant loss of population, we could actually bring them back into existence. So this is where we are proposing the UK National Livestock Biobank, and it's a system, as I say, very much based on what we've been talking about today, banking semen, but also banking skin samples alongside that as an extra measure of resilience, capturing the whole genetic profile, also working across genders. Banking everything with regeneration in mind. So I've spoken about how we bank skin samples and then we're doing the cell culture to make sure they're capable of creating living cells and then one day living organisms from that material that we bank. And taking a sample, so one of the samples we're going to work with, obviously still promoting very much semen collection, but also utilising the fact that our livestock, most of them need to be ear tagged, so we can use that little punch biopsy there to preserve the skin in that way from the live animals, and that's what we were doing on the country file. And collection of semen, so different, so for the livestock sector, it's quite heavily dominated by the big commercial companies, banking down semen that's relevant to our production requirements right at this present moment in time. But actually in the future we might find that we need a more robust and wider resource of semen. So another one of our goals is actually to collect on-farm frozen semen, those that aren't heavily represented in the food chain system, and bank them for further diversity in semen samples in the future. So diagram basically summarising the applications, and it's the same as what we've gone through today for the rare breed horses. So genetic replication, assisted reproduction, genomic passports and future technologies, they're two different things for the livestock. But genetic replication, so it's the same diagram, same principle, but just applied to different species of banking what's available to us so that we've got resources to go back to upon a population narrowing in the future. Assisted reproduction, as I said, with induced pluripotent stem cells, so that we can bank these skin samples and not just be limited to creating genetic twins, we can actually create fresh genetic sets. And that's an example of mouse pups created from stem cells generated from gametes. Genomic passports, so that's slightly different. For the livestock, we envisage a future where they will be required genomic passports, so at the same time they're banking material for biobanking, DNA can be sought for the passport too. The last application though, and this is applicable all across the species, and it's this quote I started with, is collecting things for reasons you don't yet understand. So the reason why we want to keep that initial skin cell preservation, not just taking it, putting it in the tank for future use is low cost, is because if we want it to be widely accessible to people, so that even if you're not sure what we're going to use that resource for at the minute, it's at least just banked. These animals are here for us one time only, so we really need to capture that DNA while we've got it with us and just get it banked for if and when we require it in the future. And I think this, it's on the livestock biobank presentation, but it's applicable to everything we spoke about today, really. Once a breed goes extinct, its genetics are lost to history, genetics that farmers may need in the future to combat outbreaks of disease, a changing climate, or genetic issues that arise from livestock being too closely related. We must act to preserve and safeguard what is available to us today to ensure a sustainable, resilient, and adaptable tomorrow. So I think that really just summarises in points all that we've spoken about today about collecting diversity of resources to future-proof ourselves and provide for breeds in the future. And that's the end of my presentation, and it's on time as well.